Hello learners, I welcome you to this module on selection of microorganism for the process of fermentation. Microorganisms are one of the most important part of a successful industrial fermentation. The selection of microorganism should be based upon various factors. The most important factor is the production potential of the strain selected for fermentation. The production strain which is selected for industrial fermentation should satisfy the following characteristics. It should be high yielding strain. The organism should have a stable biochemical feature. This strain should not be able to produce any undesirable product. The cultivation conditions should be easy to maintain. The screening techniques. Detection and isolation of high yielding species from the nature as a source material like soil or water is called as screening of microorganisms. The screening is performed at two levels: the primary screening or the preliminary screening and the secondary screening. The primary screening consists of very basic examinations of microorganisms to qualitatively detect and isolate the organisms which have the ability to produce the desired product. Primary screening is performed to find out whether the organism can produce an antibiotic, an enzyme or a vitamin or any other economically important product. The primary screening methods are qualitative examinations of the organism, whether it has ability to produce the desired product or not. The test is generally carried out in a plate and therefore it is called as plate screening method. There are some special methods to perform successful screening of the organism: the crowded plate technique, the Auxanography, enrichment culture technique and use of an indicator dye. The crowded plate technique. This is a special method designed for screening of organisms which have ability to produce antibiotics. Dilutions are made of the source material, that is soil or water sample, and then spread on nutrient agar plate following the spread plate technique. The inoculated plates are incubated. The plates having more than 300 to 400 colonies per plate are considered. Since it is very easy to find out the colonies having antibiotic activity in an overgrown plate, those colonies which have antibiotic production ability are easily detected because around them no other organisms grow. Such a colony which keep itself isolated amongst the crowded colonies is then selected for detection of antibiotic production. The zone of inhibition seen in the crowded plate around a colony is an indication of antibiotic production or an inhibitory substance by that colony against the other unwanted organisms. This method was improved later by using the test organism modification method. This technique has a modification that agar plates which are well grown colonies between 100 to 300 per plate is inoculated and flooded with the suspension of test organism. These plates are subjected for further incubation to allow the growth of test organism. The formation of a zone of inhibition around the colony indicate antibiotic activity. The diameter of zone of inhibition is measured in millimeters. It is an approximate of the quantity of antibiotic produced by the various colonies. Those colonies are selected having larger zones of inhibition which are purified and further tested. Auxanography. This is a special method which is designed to detect the organism which have ability to produce growth factors like vitamins, amino acids, growth factors in the medium. The two major steps involved in this method are preparation of the first type of plate and preparation of the second type of plate. Preparation of the first type of plate. A filter paper strip is first taken and put in the bottom of the Petri plate in such a way that the two ends pass over the edge of the Petri plate. A filter paper disc of the size exactly equal to the bottom of the Petri plate is placed upon that filter paper strip at the bottom of the Petri plate. Nutrient agar medium is then poured onto the dish having the filter paper at the base and allowed to solidify. The microbial source like soil is then diluted and appropriate dilutions are spread on the plate, which will produce isolated colonies. Preparation of the second plate. The minimal agar medium, which is lacking in that particular factor for which we are considering this experiment, is seeded with the test organism. The seeded medium is poured onto the surface of the fresh agar plate. The Petri plate is allowed to set. The Petri plate, which is prepared in the first step, is aseptically lifted out with the help of the filter paper strip and placed on the surface of the second Petri plate as prepared. The growth factor producing colony is present on the surface of the first layer of the agar, which can diffuse into the lower layer of the agar containing the test organism. The zones of stimulated growth of the test organism around a particular colony is an indication that it produces growth factors extracellularly. The production colonies are subcultured and further tested. Enrichment culture technique. The technique was designed by Bejernik in the soil microbiology to isolate an organism from the available population present in the soil having desired characteristics. Either the medium or the incubation conditions is selected such that it would favor the growth of the desired microorganism. The conditions selected automatically inhibit the growth of the unwanted or the undesirable organism. It consists of following steps. For example, if you want to isolate an organism producing a amylase enzyme, then the media which is used for isolation and growth is a starch agar medium. In this medium, starch is the only carbon source available. There is no other carbon source present in the medium. When the soil samples after proper dilutions are plated on the starch agar plate, then colonies appear after incubation. These colonies of organisms show that they can use starch as a source of carbon. That is, they have the ability to produce amylase. Iodine is then added upon the grown agar plate and a zone of colorless zone is observed. A colorless zone is observed which is called as the zone of hydrolysis around the dark ink blue background because iodine reacts with starch and gives a dark blue color. The colorless zone indicates hydrolysis of starch. The clear zone around the colony indicates the production of extracellular amylase. Use of an indicator dye. The indicator dye is a molecule which has the ability to produce color and change them with a change in the pH. Therefore, they are excellent indicating systems used in screening of organisms which produce organic acids or alkalis. The change in the pH causes the change in the color of the dye. The commonly used indicator dyes are neutral red and bromothymol blue. The medium which is taken for isolation is a poorly buffered nutrient agar medium containing the indicator dye. Change in the color of the colonies of the cells which grow on the medium indicate production of organic acids. The color change in the cells also indicate and require further testing to identify which kind of acids are produced by the microorganism. I hope this module has helped you to understand the first step of screening of microorganisms, that is the preliminary screening, which helps you to understand whether a particular microorganism has potential for production of a particular product of interest. Thank you.